Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, dear fabulous students. Um, I want just to tell you that I'm very happy that I'm meeting you again, as I miss to hear from you all. Honestly, I want to start with a big advice to you all as a biology teacher, as your father, as your friend, whatever you think about me. Please be careful of yourself. Be careful. The government, the education administration, all of us, we plan to give you a home set because we are caring about you. This is meaning that you have to understand that you are so important for us. You are so important for your society. You are so important for your nation. You are a good citizen, and we are really caring about you. So my advice to you at the beginning, before I start my session, that please take care of yourself. Eat only healthy food. Stop eating these artificial things. Stop eating this food that is negative energy. Like these chips, what you are eating, like this Indomie, what you are eating. Try move to the health way. Because this virus, what we have right now, you could look at it as if it is really a global crisis. So please be careful of yourself. Eat only healthy food because this virus is killing the body when the immunity is down. What can make the immunity down? Only the artificial and the unhealthy food. Please go ahead and be careful of yourself. Today we are going on to start with our session in biology. We'll continue from the point where we stopped. On the previous times, we started already with mammals, and deep in details about mammals, how we classify it into mammals, what are the types of mammals, what are the characteristics of mammals, what is the behavior of these kinds of mammals, what are these kinds of behaviors, how it's looking like, what's going on. I think we covered all this, and I feel satisfied that I see you all understand my sessions from before. Today, we'll continue in this amazing field while moving on this way. Today, our subject and our title is first lesson of chapter 12, which called DNA. DNA. What? DNA. What does mean DNA? One of you could think of DNA is the material stuff inside the nucleus that is given the natural or given the traits and inherited traits for humans. Wow, it's amazing. It's a good idea. But please, this is not, it's not scientific way to identify DNA. One of you could tell me, I know what does mean DNA, Mr. Ashraf. What is DNA in your opinion? He could tell me that DNA is deoxyribonucleic acid. You heard it like this. You read it like this. Please. There is big difference between di and D in scientific language, which is coming from Latinic language. Di means double. But D means mist. That does not exist. So when I'm saying deoxyribonucleic acid, this is meaning that it is two strands. And this is not fair. But it's not meaning this. It's meaning deoxyribonucleic acid. What? Deoxyribonucleic acid. DNA. It means what? Excuse me, Mr. Ashraf. So what is the difference between deoxyribonucleic acid and RNA? We say deoxy, just avoid this two word, this part, okay, this prefix. Avoid this prefix from this word. So it is ribonucleic acid, RNA. You understand? So only the difference between RNA and DNA is something related to oxygen. That there is no oxygen. There is lack of oxygen in the DNA. But in RNA, it exists. Okay, we will not come deep in these details right now. We will come deep in these details soon. Okay, here. Biology for grade 11, chapter 2, deoxyribonucleic acid. There are some essential questions. Do you remember what I explained to you, dear fabulous students, when we said, what is your opinion by the end of the week about K, W, L, K, W, L? Hussein, please, attention. It's very important for you. Haji, attention. All my colleagues, all my students, please, attention. K, what do you know about DNA? W, what are the questions in your mind? What you are thinking about? And what you want to feel satisfied by understanding it? And DNA. L means by the end of the session, by the end of the chapter, by the end of the week, what information, what knowledge you gain. This is the most important for me. Okay. Now, we are going on to know or to read these questions. Which experiments led to the discovery of DNA as genetic material? Yes, we have to think well. How the people, how the scientists understand and know that DNA is important and it has the whole information about the cell. How it is controlling the whole cell. How it is creating and doing inside the cell. What is the activity? It's very important. How the scientists discover this? Name some of these scientists. Because what we already know about Mendel, Gregor Mendel. What's next? Well, going on to come deep in these details. Okay. The second question is the essential question, very important to all of you. What is the basic structure of DNA? Wow, what is the basic structure of DNA? How it's looking like? How this DNA looking like? What's going on? It's very important. The third question, what is the basic structure of eukaryotic chromosomes? What? Now we have several words. Yes, we have heard about these words. About DNA, we have heard about it. The structure, the basic structure of DNA, maybe some of you know some information about it and some of you not. Okay. What about the basic structure of eukaryotics? Chromosomes. This is very important for you to know what is the structure of eukaryotic chromosomes, what is chromosome. By the way, as you know me, I'm always connecting your way of life, I mean your religion, your Islamic religion, your Quran, with what we are discovering in the nature, what we are discovering in the science field. By this way, this will make you more proud of yourself that you are Muslim, more proud of yourself that you are doing worship to your God. Let's come to know this information together. By the way, we have some words, you know it from before, like nucleic acid. And the new words, what you should know is the double helix and what is meant by nucleosome. Okay, here, we're going on just to discuss briefly, I will not come deep in details, okay, about the discovery of the genetic material. Okay? The discovery of the genetic materials. After the rediscovery of Mendel's work, I think you remember very well what happened about Mendel's and Drosophila and what's going on and everything about this. Well, I don't want to be back to it because really you are, mashallah, you are so smart and you remember this because you studied it with my colleagues, my subject in the previous years.
Scientists began to look for the molecule involved in inheritance.
That's true. That's true. Why? You are looking similar to your dad. Sometimes your brother is looking similar to your uncle of your mom. Why? Why? What is going on? What had happened? That is the most important point we're going to discuss. For many years, scientists struggled. Yes, there was a struggle, a real struggle. And I don't want to tell you the stories about this. It's too much stories, okay? To determine if DNA or protein was a source of genetic information. What? What? DNA or protein? OMG. So what is the relation between DNA and protein? Here we have to stop and think well. What is the relation between DNA and protein? Oh, I will just tell you one thing. Remember this, please. There are codes. I write it now. Okay. Remember it. A, T, C, G. This is with DNA. Remember this. A, C, G, U. What? A, C, G, U. This is adenine, thymine, cytosine, and guanine. Here is adenine, cytosine, guanine, and uracil. Uracil. So where is thymine? Thymine is replaced with uracil. Okay, think about this and we'll discuss it deeply later. Okay, in the next session. Okay. Griffith. Griffith is a scientist that he made some experiment to check what's going on. First major experiment searching for the genetic material. Okay. Remember this, I'm not coming deep in details. I'm not discussing the procedures what he made. Okay, just step by step, just briefly I'm telling you. Okay. Involved transformation between two forms of S. pneumonia. This is a kind of bacteria. Set the stage for the search to identify the transforming substance. So, look what he made. Look at this picture. Look at picture A. Look at picture A. Here, what he made, he injected the mouse by what? By S. S means smooth, strain. This disease causing. This smooth strain is making disease. It's like this, there is a smooth and there is a rough kind of this bacteria, pneumonia. Okay? So he injected it by smooth one that is really active and producing toxin in the blood. So the mice die. The mouse dies. Okay? Here, he took the rough one off, means rough strain. It's not diseasing. What? It's not diseasing. You mean that the animal or even human can be infected by a kind of any of these infectious microorganisms, and then he is okay and he's not feeling anything and there is no symptoms appearing on him? Yes. Really? Yes. This is why it is really dangerous to be infected by corona nowadays, because from the very beginning you couldn't feel anything, you are feeling normal. But after 2 weeks, the symptoms started appearing, but it is coming rough and tough. Why it's coming rough and tough? I think I'll discuss this with you next session about corona especially, okay, and the influence on the next session, inshallah. Okay, so the mice, everything was okay. So what they made? They heat kills a strain, the smooth strain. Then they injected it to the mouse stay alive, he's not dying. Wow. Look what's going on here. They mixed heat kills strain with our strain, rough strain, and inject it to the mouse, it is died. Oh my God. Okay, this is what Griffith made. Okay. Avery identified the molecule that transformed the R strain, the RAF strain of S. pneumonia into S strain. Concluded that when the S cells were killed, DNA was released. What? DNA was released. All this is just conclusion. You see? Concluded that when S cells were killed, DNA was released. Left the cell. So what's going on? R bacteria incorporated this DNA into their cells and changed into a cell. Oh my God, so it is a kind of modification, okay? It's developed itself, okay? DNA, you know, you could say it is a kind of mutation, you know? Have you heard about mutation? Yeah, mutation is change in DNA that changed the traits that's appearing. Okay. Okay, next. Diversity of the genetic material. This is about two scientists, Hershey and Chase. They used the radioactive labeling to trace bacteriophage. Oh my God. Bacteriophage, I think I discussed it with you from before. Bacteriophage is looking like this. And here exactly there is DNA. Look at this picture, dear students. The group A and group B. Look at these two scientists, what they made. Here, viruses are grown in a medium containing P32 to lab DNA. And group 2 viruses are grown in medium containing S35 to lab protein. To lab protein. Here to lab DNA. Label DNA and label protein. Okay? What happened here? What had happened here? Let's come to check what's going on. Escherichia coli and viruses are placed together into liquid culture medium. Both of them, this bacteria and this bacteriophage. By the way, bacteriophage is, it is looking like the ring or the edge between viruses and bacteria. Okay, here viruses infect the bacteria. Oh my God, injecting their genetic materials, injecting it inside the Escherichia coli, the E. coli, injecting it inside. Okay, but here in the medium of DNA. And there, exactly, it is in the medium of phosphate 32 to label DNA. Here, in medium containing sulfur to label protein. Okay. The mixture is, look what's going on. The mixture is agitated to dislodge the viruses from the bacteria. Then what is the result? Look here. Viral proteins, radioactive bacterial cells. There is a viral proteins here. Viral proteins. Viral related to viruses. Proteins. So the protein of this virus. You got the idea? Okay. Here there is a radioactive viral protein as the same. But bacteria cells, what was here? And here, bacteria cell as well. So what's going on? The bacteria are separated from the liquid containing the virus. So, virus injects its DNA, just attached on the surface of this bacteria, of this Escherichia coli, then inject its DNA. The DNA inside made a replication. So there was a replication. So it killed the DNA of the bacteria and replaced it by its own DNA. So bacteria became like a sack of group of bacteriophage, this kind of viruses. Okay? That's okay. Wonderful. Please, dear students, about this part, I want you to go to your connected resources to play interactive table summary of Hershey Chase results. Please don't forget to do this and list it from your account. Actually, I don't want to come deep in these details right now. This is more than enough for you. Next, we will continue, inshallah, by the next session of the beginning of next week. Please go ahead, and I really miss to hear from you all. Contact me if there is anything you want to know it more, and good luck, dear students. Thank you very much.